This course is a collaboration between Faculty of Tropical Medicine, Mahidol University, and Wellcome Trust Sanger Institute. It provides a hands-on sequencing training for the students and potential postdocs that are going to do research in the AMR, which is shorted from antimicrobial resistance. The hands-on part is really important because sequencing and analytical data, you cannot just learn by the YouTubes or reading the manuscript or the commands. Having the expert from around the world to come and guide you, advise you on how to do the coding, analyzing the data is really helpful. Antimicrobial resistance is a big problem in the whole world, particularly in the low middle income countries. Sequencing and how to analyze the bacterial sequence data can be a powerful tool to reduce the burden of the AMR. It can lead to the better actions in the hospital to stop the outbreak or the clusters of the AMR infection. Obviously, translated the skill into the action is still a long road to go, but then you need more manpower and people to be able to do it and go back to their own country, set up studies or supporting the Ministry of Public Health and their own hospital actions so that when they have the cluster or the outbreak, they can do it. So that lead to the better hand hygiene or improve the cleaning of the hospital or the antimicrobial stewardship. It can also go to support the research that they are doing, having the better understanding of the AMR in all low middle income countries, and that will support the people, Ministry of Public Health, and more funding into their own hospital and their own countries. For the impact, I hope that in the long term we can save many lives with this course. But that's a long term, right? But then in the short term, we hope that the participants will have more confidence in their own skills, in their own analytical mechanisms, teams, network, consultants from the global experts, so that they can go there to their own hospital, start or continue their own research, make it bigger, going to the world-class research, and also to support the actions for the AMR in their own hospitals and their own countries. With that short term, we hope that they can improve to be the global leaders and their own local leaders for the AMR and have the impact to the Ministry of Public Health on their own country. Although yes, that's a long term, but that's the impact that I hope that short term and they can carry on to grow up to make the mid-term and long-term impact. And at the end of the day, more lives can be saved from the AMR.
Yeah, so this course focuses on antimicrobial resistance in bacterial pathogens, and the idea of the course is to provide the participants on the course a kind of an end-to-end view of the process of using genomics to understand antimicrobial resistance. So there's a laboratory component where the participants follow testing procedures, phenotypic testing procedures. They look at quality control in the laboratory, and then they move into genomics where they're analysing genomes, starting with the raw data, assembling the data, building phylogenetic trees, and then interpreting the data, looking at using computational methods to identify resistance mechanisms. So it teaches them the entire process of using genomics to understand resistance. Well, obviously AMR is a global problem and it affects countries all around the world. So this course focuses on scientists in Asia. So we have scientists from all across Asia who are already working in antimicrobial resistance. So I think the course has two particular aims really. One is to provide training where there may be gaps in individuals' knowledge, particularly if they're quite junior researchers, but also two, it aims to bring together researchers from across Asia and build new collaborative networks. Well, we hope that the participants find it useful. It's quite common that certain parts of the course are useful for one participant and another part of a course is useful for to a different participant depending on their background. So we hope really that they just learn something that they can take back to their own work, but also that they feel empowered to see how they could use genomics maybe in a more focused way in their own research, but also that they build these networks and contacts across Asia that hopefully will be useful for their future research in AMR.
In this workshop, I assist in the laboratory, two laboratories. First is the antibiotic resistance test. We use the E. coli and Staph aureus to take the antibiotic resistance. And the other lab, I will use the nanopore sequencing to do the sequencing of the gene resistance of that E. coli. This technique will help us to know which gene of the E. coli or plasmid that make them resistant to the antibiotic, so we can know in the future how the evolution of this gene transfer to the other bacteria or cause disease in the environment or in the patient in the future.
In AMR course, I take responsibility about the lab part in the topic of antimicrobial susceptibility testing, or you can call like the short AST, and the technique of broad microdilution test and the broad microdilution for meropenem. This technique can use as the screening test in case of once you have like the one isolate and you want to know that the isolate is susceptible, resistant or intermediate. You can use this technique for screening, and for the broad microdilution, you can use the broad microdilution to confirm that the organism that you got is susceptible, resistant or intermediate. And this one can be used like give the result to the doctor, and they can maybe apply or maybe adjust for the drug treatment for the patient.
I was actually one of the attendees in AMR 2023 here in the same place and the same training team, and I was fortunate enough to be invited by the Wellcome Kinetic Science team to be here again to be part of the training team. I guess one of the key topics that I'm going to deliver is about a case study of the Asian genomics project that I'm involved, where I already have my sequence before the training in 2023, where I was able to apply the techniques and also discussing my projects with the training team to now to have it published. Yeah, definitely ask question, take the initiative to discuss about your existing project, like how you think you could apply the knowledge, especially genomics, with the training teams here. And then also, it's never too small to start a project, like in Asia, and think of how you could apply genomics to answer your research questions. Yeah, good luck.
Right, so for this workshop I teach some basic concepts in phylogenetics and how to reconstruct a phylogeny from the sequencing data. So phylogeny is a very good tool to depict evolutionary relatedness of organisms, right? And then you can use it as scaffold and map multiple layers of information onto this scaffold or family tree, for example, like the AMR profiles. And also the country of origins of the isolates, for example. And from then you can use the information to reconstruct transmission history of drug-resistant bacteria, and to find out the origin of the bacteria, for example. And from that you can maybe use those information to better control the spread or the outbreaks of the AMR bacteria.
The module that I will be handling in this workshop is metagenomics in AMR detection. So this involves using reads, particularly Illumina short reads, and doing downstream analysis in terms of identifying the AMR genes from the sequences, and also functional annotations and the taxonomic classifications that we can get from the metagenomic data. And this will help in getting a global understanding of what are the organisms which are present in the sample, what are the antimicrobial genes that are present, and how these can be used to derive insights. So these tools are very helpful. The tools that I'll be teaching are very helpful in identifying specific antimicrobial resistant genes. This is particularly because we will be dealing with taxonomic identification, functional annotations, and characterizing different AMR genes from the sequences itself. And when you look at it on a global scale, the techniques that I'll be teaching in the lab will basically be used in global surveillance of AMR genes from multitude of settings, including hospitals, wastewater, and so on and so forth. And this helps in the global understanding of AMRs, especially integrating into the One Health framework, which I think all countries are into, trying to identify AMRs from different scenarios.
I taught about read mapping and variant calling, where you take sequence reads from your sample or somewhere else and align them with a known reference genome, and then call for variants or differences between them. This can be SNPs or single nucleotide polymorphisms or indels. So read mapping and variant calling are crucial for AMR research because it enables us to detect AMR early, understand the resistance mechanism, and even guide treatment strategies. Both of these techniques are very important in surveillance of AMR pathogens, and even in updating AMR databases, which can aid with our research. So both of these techniques support real-time surveillance, track AMR evolution and contribute to resistance databases, which aid in better policy making and antibiotic stewardship.
Yeah, there are many interesting things that I learned a lot in these 2 days, and I am expecting in coming days, such as the AMR was one of the most important thing. There are many tools, there are many other things that were part of this course which interest me, and I really learn a lot, and I also learned some new techniques to detecting, to visualize and to profile these AMRs from this course. Yeah, the most important thing, the part of this workshop is actually the wet lab. Yeah, I forgot some of the techniques when I was in my undergrads, and then after 3 to 4 years I came back to the wet lab, and it's really fascinating and really most fascinating thing and the part of this workshop.
So the best part till now I learned about NGS, and also in upcoming session I am looking for the metagenomic session so that I can learn both of them together. Yeah, so currently I am working in structural biology, and so if I able to learn the genomics part, especially in NGS, so in future it will be helpful for me to know the mutations and do the variant calling, which is very helpful for my research. So that's why I'm here. Actually it is really good, and the thing is I am like the best part that here it is like I am doing the wet lab and also the dry lab, so it is a combination of both. So hands-on is very important, and so here I am getting that opportunity to learn the hands-on and then do the dry lab as well. So that's the best part ever I have here, so because I am getting the hands-on training.
Acquiring knowledge of genome sequencing is really important to me because it's helped me to track AMR, and because working on both field, clinical and lab department, I see that AMR is really important and a serious and growing challenge in Vietnam and also across the world. So that's why I really want to know how to track AMR, and I hope this will help me to improve infection control in my country. Yeah, I really enjoy class of bioinformatics because it's easy to follow and supportive due to instructor. They really friendly and really good. So and I feel that I'm now quite confident to conduct again bioinformatics when I come back home. Yeah.
Okay, I think the components of this workshop are excellent, but my major learning from this workshop would be genotypic and phenotypic assays for genomic prediction, and the fact that we are learning it right from the bottom and doing everything using real samples. I think that is the best part of the workshop. Number one, I made new friends and met new people. I'm exploring the city with new people, and also the fact that excellent trainers from excellent places are here and they are training in such a nice way, and they're so patient and cooperative with us. It is very smooth. Yeah.
So in my lecture and the practicals that I'm working on, we explain the genetic basis of AMR. We explain how AMR has a genetic basis and give them the knowledge they need to interpret genomic predictions of AMR. We know that it can be tested in the lab using phenotypic methods, and increasingly we are using more whole genome sequencing as a tool to detect AMR. We give them bioinformatic knowledge, basic bioinformatic skills to run these tools on the command line, and also present them with alternatives to run them on web-based tools. And we put an emphasis on interpretation. It's not always easy to digest, distill all this information that comes out of bacterial genomes. So we give them tools to compare the results between different tools and databases, keep a skeptical eye, a critical attitude when analyzing this data. I think the key message is that antibiotic resistance is a growing problem, that there's a variety of methods, phenotypic methods we use to test them routinely in the lab, that we expect in the next years to use genotypic methods increasingly, and we want them to be able to interpret those predictions, those detections with high confidence.
From this workshop, I learned a comprehensive understanding about the antimicrobial resistance and its challenge at the global setting. And in this workshop, it provides me intensive training about the latest methodologies on sequencing and also bioinformatics to let me use in my research and work. And it also allows me to have collaborations with the speakers, trainers, to have a good collaboration in the futures. Indeed, this training provides good information and knowledge and skills that I can use directly in my research and work. For example, in the One Health antimicrobial resistance, to fight the antimicrobial resistance at the global settings. And it also provides me a very good platform to integrate with the speakers, with the trainers, and also the fellow participants to have a good collaboration in the futures.
The one thing that I really want to learn from this course is actually the bioinformatic parts, when we do the downstream analysis using all the bioinformatics things, because I really have few knowledge about that. Okay, for my project, I'm focusing on these bacteria E. coli and Klebsiella that are producing ESBL. So basically, I'm going to analyze their genome. So the bioinformatic parts really play a big role in my project. So I'm going to use that definitely when I'm going back to Malaysia.